Hello learners, welcome to NR Studio. This is live Udhya program for Science and Technology, secondary course of NIOS. I am Neelam Gupta, course coordinator of Environmental Science. Welcome you all in this program. Learners, we have discussed the lesson number 24 in reproduction in our previous programs. That time we have discussed about what is reproduction, different types of reproduction, that is asexual and sexual reproduction, different types of asexual reproduction and various types of vegetative reproduction in plants. It can be it may be natural as well as artificial. And we have also discussed about sexual reproduction in plants and in humans. We have given an idea about ever increasing population and controlling measures too. And during next program we have discussed about RTIs and STDs, which is caused by the microbes, and we have also shared information about our utilization of making safe informed choices. And now we can talk in general. When we talk about reproduction, means growing babies, and we know that human babies looks like human beings and also resemble closely or distantly the parents, grandparents, or even cousins or uncle and aunt. So sometime one thing also came our mind, why is a puppy or kitten looks like a miniature dog or cat to look at? How does it possible? Or when we talk about plants, one question came to our mind, that why does a seedling acquire the same kind of leaves, stems, flowers as the parent plants? So this is all because of heredity or genetics. So science of heredity and variation is termed as genetics. So we will discuss lesson number 25, heredity of the module 5, the living world, during this program. So during this program we will discuss various aspects of the heredity. So now we will talking about objectives of this program. So first we will define what is heredity and variations, pattern of Mendelian inheritance, location, structure and function of chromosomes and genes, and briefly discuss about DNA fingerprinting and its significance. Next we will talk about process of DNA replication in brief, blood groups in humans and the manner of their inheritance, and chromosomal basis of sex determination in humans. All are we discussing in very brief. Certain hereditary disorders and also hazardous cousins, marriages, emphasis of the relevance of genetic counseling and human genome, and some salient features of genetic engineering too. So before we start this topic to discuss different aspects of the related to heredity, first we will introduce few words. That is inheritance. Inheritance is the process by which characters are passed from parents to their progenies. Heredity is a tendency of progenies to resemble with their parents, and variations is a tendency of progenies to differ from their parents, and variations are due to genes or environment. So we can say basis of the heredity is physical basis, that is gamete, and chemical basis, that is factor or genes. So first we are talking about heredity is the passing down of characters from parents to offspring is termed as heredity, and it is controlled by genes. So we can see to observe, or we can say some similarities or differences. How can we observe? So look around and you shall find there are so many differences between individuals of the same kind. For example, we can take rose garden. The color of rose flowers on a different plants are different, and puppies of the same mother dog are different in their color code, means color of their hair or fur. So all such individual differences are termed as variations, and it is due to genes or environment. You can perform or you can show through an activity to find variation in the human being. So, we take example earlobes of the human beings. Check your earlobes and those of your friends and family members too. So, what do you find? So, lower end of the earlobe may be attached or free. This feature of the earlobe is hereditary. So, you can find you can observe earlobes of your parents and your siblings, brother, sister, and note from which of your parents you have inherited this feature. You may similarly try and observe the rolling of your tongue, and you can notice who all in your family can roll their tongues. Similarly, you may not note that who all in your family can curve the tip of the thumb backward and who all cannot. For this ability is also we can say hereditary. So, you can note any other differences or through this activity, like color of the eyes or the shape of the nose or any other feature among your friends too. So, all these activities can do by yourself, and you can observe and find the differences and similarities. So, differences that you may note are termed variations, and you may also perform other activities to find out about the occurrence of the variations for yourselves too. So, variation with respect to the ability to rolling tongue or having the hitchhiker thumb. So, question about the heredity intrigued many scientists of years, but work of the Mendel was being recognized. बहुत सोचा लेकिन कुछ लोगों का ही काम रिकॉग्नाइज हुआ, जिसमें मेंडल एक थे. सो वी विल डिस्कस इन ब्रीफ व्हाई वी से Gregor Mendel as a father of genetics, or why is Mendel considered as the father of genetics? The reason is this: he had initiated work on heredity or genetics, so he has propounded laws of genetics. So, first we will talk about contribution of the Gregor John Mendel. Gregor John Mendel, born in 1822 and died in 1884, an Austrian monk, under the laborious task of finding the answers, he selected some pea plants, grew them year after year, compiled a lot of data, analyzed and postulated certain laws of inheritance. For the first time, father of genetics, for discovering the fundamental laws of inheritance through his meticulous experiments with pea plants between 1856 and 1863. His remarkable work, however, got recognized years after his death, when Correns, Trestmark and Hugo de Vries came to the same conclusions as Mendel did, after independently carrying out experiments in their own countries. So, now we will talk about the Mendel's law of inheritance. Mendel's laws state that every feature or character, like color of flowers, height of the plant, color and texture of the seed, color and texture of pod, and location of flower on the plant, is controlled by the pair of factors. During the formation of gametes, one factor goes to one gamete and its pair to another gamete. Thus, the two factors of a pair segregate or separate during gamete formation. So, upon fertilization, the combination of factors expresses the feature. That is first law. Out of the two factors controlling a certain features, dominant one may express in spite of the presence of the other. The other factor expresses only in the absence of the dominant factor and is termed recessive. That is second law. For example, factor of tallness in a pea plant always expresses in the offspring, but roughness expresses only if factor of tallness is not present. So, Mendel also postulated two other laws called the law of parental equivalence and law of independent assortment. They are not elaborated here. First law defined as universal. Scientists later observed deviations from the other Mendelian laws. Sutton in 1902, working with grasshopper chromosomes, confirmed that Mendelian factors were present in chromosomes. Still later, the term gene replaced the term factor. So, genes are present on chromosomes. So, genes are responsible for heredity and they are present on chromosomes at very fixed point. So, now we will talk about chromosomes and genes. Chromosomes, the nucleus of every cell, contain a fixed number of chromosomes, and in all the cells of eukaryotes, chromosomes show typical characteristics as they are present in pairs, one from the father and another from the mother. They can be seen only during cell division. In a non-dividing cell, they appear in the nucleus as a jumbled up network through termed chromatin. Paired chromosomes are present in a fixed number. A fixed set of chromosomes is termed as diploid or paired number and designated as 2n. Each chromosome is made of one molecule of the chemical called DNA or deoxyribonucleic acid and some proteins. Before cell division, DNA molecule of a chromosome replicates to give two molecules of DNA which are called chromatids, and the two chromatids of the chromosome remain attached at the point called centromere and separated to form two chromosomes during cell division. In a bacteria, only one chromosome is present, and since there is no well-formed nucleus, the single chromosome lies in the cytoplasm in the region called nucleoid. First figure A shows bacterial chromosome and second figure shows eukaryotic chromosomes. Now we will talk about human chromosomes. We know every cell of a human being consists 46 chromosomes. Diploid number in human is 46. This can be expressed as 2n is equal to 46. So, since gametes contain only half the number of the chromosome or the haploid number, a sperm and a ovum or egg has only 23 chromosomes. So, we know every species has a fixed number of chromosomes. Each chromosome seen in a figure possesses two identical chromosomes joined by the centromere. Chromatids become independent chromosomes when they acquire a centromere at anaphase of the cell division and ultimately move to different cells. So, chromosome may be photographed from dividing cells at metaphase stage of mitosis and then displayed in pair as in the figure. A pair of similar chromosomes, one received from the father and one from the mother, containing the same genes, are termed homologous chromosomes. Out of the 23 pairs of the chromosome, 22 pairs are termed autosomes, and 23rd pair, XX in females and X and Y in males, are called sex chromosomes. X chromosomes has several genes, some of which are necessary for survival. But Y chromosomes bear genes for maleness only. So, one such gene is the testes determining factor. So, genes are present on chromosomes. The genes are the Mendelian factor present in pair on the chromosomes. We have already told. So, one member of a pair of genes present the chromosomes has its pair on the homologous chromosomes at the same location. So, genes are the bearer of the hereditary characters or the units of heredity. A chromosome contains one molecule of the called DNA. Genes present on chromosomes are segments of DNA, and since every individual begins life as a single cell, so DNA contained in all cells of the individual is identical. So, you might have heard that DNA fingerprinting. We are talking about, so criminals can now be identified by DNA test called DNA fingerprinting. This is because DNA of an individual is the same in each and every cell of the body and also resembles the DNA of parents. So needless to say that this is because children inherit DNA from their parents. So just like the fingerprint, DNA of every individual is unique, and even if a hair or a drop of blood or semen of the criminal is left at the site of the crime, it can be used to detect the DNA of the criminal and compare that with that of the suspect to ascertain the truth. So, do you know our Indian scientist Dr. Heredity, who works in the field of the molecular biology? So, we will talk about very brief about Dr. Heredity was the creator of man-made gene. It is a matter of pride that Nobel laureate Dr. Heredity, who was born in our country, synthesized an artificial gene in the laboratory for the first time. For this, Dr. Khurana got the Nobel Prize in 1970 for his contribution to molecular biology. So now we are talking about DNA molecule. DNA molecule is a polynucleotide. Poly means many. It is made of units called nucleotides, each of which nucleotide contain nitrogenous base, deoxyribose sugar and a phosphate. So there are 4 nitrogen bases: adenine, guanine, thymine and cytosine, and hence 4 kinds of the nucleotides in a DNA molecule. There are various combinations of these nucleotides in a segment of DNA form the different genes, and in physical structure a DNA molecule is a double helix containing 2 polynucleotide strands. So now we will talk about DNA replication. When cell division takes place in a manner so that one cell divides into 2 identical cells, that is mitotic cell division, with the same number of identical chromosomes. Therefore, prior to cell division, every chromosome should contain 2 chromatids made of 2 identical DNA molecules. So, this is achieved through the process of DNA duplication or DNA replication. So, there are several major steps in DNA replication. So, therefore, prior to cell division, every chromosome should contain 2 chromatids made of 2 identical DNA molecules. So, double-stranded DNA molecule unwinds with the help of certain enzyme to expose 2 strands of DNA. DNA polymerase enzyme catalyzes the formation of a new daughter strand, which can form a double helix with 1 strand of the parental DNA molecule. So, 2 DNA molecules with each with a parent strand and a new strand get generated. 2 identical DNA molecules then became 2 chromatids, which remain attached by a centromere. Thus, upon DNA replication, each chromosome contains 2 identical molecules of DNA housed in its 2 chromatids, and during cell division, these 2 chromatids separated out of 2 chromosomes, one each passing into the 2 daughter cells. So, every one of us is born with genes inherited from our parents. So, our blood group depends on the combination of a pair of genes, one of which is inherited from each parent, and there are 4 blood groups: A, B, AB and O. So, every human being has 1 blood group out of the 4. The genes which control the inheritance of these blood groups are designated as IA, IB and small I. So, when a fetus growing fetus means growing young one in mother's womb develops, its blood group is determined by the combination of any of the two that mentions genes, one recessive from each of the parents. So, the gene combination and the resulting blood group are shown in the table on the next slide. You can see IA, IB, IA or I. Blood group is A. Gene combination is IB, IB or IBI, small I, blood group B. IA, IB, blood group is AB. Small I, blood group O. So, now we will explain little bit about this figure. So, this figure explain the ABO blood group system and how gene combination determine blood group. Gene involved, there are 3 alleles for the ABO group gene. IA produces A antigen, IB produces B antigen and small I produces no antigen. So, dominance pattern is IA and IB are co-dominant, both expresses together, and small I is recessive, expressed only when paired with another small I. So, combination of the genes and the resulting DNA, as we have discussed, IA, IB, I and dominance pattern is IA and IB are co-dominance and small I is recessive. So, when IA or I is blood group A antigen is present on RBCs, small I does not produce any antigen, so IA dominates. Second case, IB or IBI, blood group is B antigen is present on RBCs, I is recessive, so IB dominates. Third condition, IA, IB, blood group AB, both A and B antigens are present. This shows codominance, means both alleles express equally. So, we are talking about 3 groups. Next, fourth blood group is no I, small I, blood group O, no A or B antigen present, both alleles are recessive. Apart from these blood groups, human beings may also belong to the group designated as rhesus positive Rh plus or rhesus negative Rh minus. Most humans are Rh plus, some are Rh negative or rhesus negative. The Rh plus genes is dominate over Rh minus gene. In case of any emergency such as an accident or a disease condition, blood transfusion may be required. So, only matching blood group of the blood donor can be transfused. So, why should you know your blood group? A person with blood group A can donate blood to patient with blood group A and AB. AB can receive blood from any of the 4 blood groups. O can receive blood only from O, but donate blood to all 4 groups. So, everybody should know what's your blood group. In case of any emergency like accident or disease condition, blood transfusion may be required. Only a matching group of the blood donor can be transfused. Entire human race can be divided into 4 groups on the basis of the blood group A, AB, AB and O. So, do you think further distinction made by human being on the basis of caste, creed and gender are justified? So, now we move the next section, that is sex determination in humans. So, combination of sex chromosomes with autosomes. We have talked 46 chromosomes, 23 pair, 22 is autosomes and 1 is different pair. So, these combination of sex chromosomes with autosomes determine whether the fetus will be a boy or a girl. Look at the figure. The fetus develops from the zygote, which is formed by the fusion of the 2 gametes, the male gamete or sperm and the female gamete or egg. Gametes are haploid, having only n number of the chromosomes, while the zygote is diploid, 2n. So, ova or eggs are of one kind only. They contain 22 autosomes and a single chromosome, X chromosomes. Sperms are of two kinds, having 22 autosomes and one X chromosome, or having 22 autosomes or a Y chromosomes, as you see in the figure. So, when X bearing sperm fuses with the egg, a female child results with 44 autosomes and 2 X chromosomes. If Y bearing sperm fuses with the egg, then a male child results with chromosomal constitution of 44 autosomes and 1 X and Y, 1 Y chromosomes. So, we have already discussed about cell division earlier and know at the metaphase chromosome are seen clearly lying at the equator and may be studied unique under the microscope or by taking a photograph. You can then easily identify the state that the chromosomes in a male human are 44 autosomes plus XY, and those in the female are 44 autosomes plus XX. So, it is therefore wrong to blame a woman if she does not bear a male child, as it done in some ignorant families of our country. Sex of an individual is purely due to chance, and neither the mother nor the father can be blamed. So, the Prenatal Diagnostic Techniques Regulation and Prevention of Misuse Act 1994 was enacted and brought into operation from 1st January 1996 in order to check female petty size. The Act prohibits determination and disclosure of the sex of the fetus. So, it also prohibits any advertisement relating to prenatal determination of sex and prescribe punishment for its contravention. The person who contravenes the provisions of this act is punishable with imprisonment and fine. So, we will take a little bit short break. After break, we will continue this discussion.
प्रकाशित करने राहों को आलोकित करने हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने। जीवन ये प्रकाशित करने राहों को आलोकित करने।
धन साधन समय की सीमा से विद्यालय जो मुक्त हो, धन साधन समय की सीमा से विद्यालय जो मुक्त हो।
कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो।
कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो।
मित्रों हम उठे और जागे,
मंजिल आने की ध्वनि सुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
ज्ञान ये प्रकाशित करने, राहों को आलोकित करने।
शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर
शिक्षा और परीक्षा का अवसर एक बार ही क्यों मिलता फिर दुनिया में इसके केंद्र बहुत। कोई मन चाहा केंद्र चुने, हम अपना जीवन स्वयं बने, हम अपना रास्ता स्वयं चुने। कोई मन चाहा केंद्र चुने, स्वयं बने, हम
Before taking a break, we have talked about what is heredity and variation. We have talked about Mendelian's law, 2 laws of Mendelian law. We have talked about DNA fingerprinting. How can DNA fingerprinting helps to identify criminal? We have talked about blood group. There are 4 blood groups: A, B, AB, and O. We have talked about how can sex determination in human does. And now we will talk about heredity disorders. So we have already given an idea about that genes controls all features of an organism. Sometimes a gene may change or mutate either in a gamete or zygote. So mutated gene may not remain normal. So also sometimes a defective gene present in the parents may not be expressed in the parent as the dominant normal member of its pair, may mask the effect of the defective genes. But if the child inherits the defective gene from both the parents, the presence of the defective pair of genes has a harmful effect, and such a disorder is known as hereditary or genetic disorder. So look at the figure. This figure, a symbol plus means normal or functional gene, minus means defective or mutant gene. A person with plus minus as a carrier appears normal because the normal gene masks the defective ones. The person with minus gene is affected or genetic disorder. Parents means mother, normal mother, genotype is plus minus. Normal father, genotype is minus plus minus. Both parents look normal, but the carrier of the recessive defective gene. So gamete, each parent produces 2 types of gametes, one plus and one minus. So offspring combines when gametes combine 4 possible genotypes, that is plus minus normal, plus normal, plus minus normal, but carrier so plus minus normal carrier and minus abnormal or having genetic disorder. So out of the 4 children, 75% children are phenotypically normal, 25% completely normal, 50% carrier, and 25 out of the 1 out of the 4 children are affected, minus. So important takeaway is this: the diagram shows why 2 normal looking parents can have a child with a genetic disorder. If the disorder is autosomal recessive, albinism, fibrosis, cystic fibrosis, sickle cell anemia. So there are several kind of hereditary disorders, some of which may be caused due to presence of only one defective gene which is dominant, or sometimes by the presence of two defective recessive genes. So genetic disorders cannot be cured by medicines. Scientists are trying to discover methods by which a defective gene occurring in an individual may be removed or replaced by a normal gene. This is called gene replacement therapy. And there are we are discussing three genetic or hereditary disorders. These are common: thalassemia, hemophilia, and color blindness. So we will talk about little bit about first thalassemia. Patients suffering from this disorder are unable to manufacture hemoglobin, the pigment present in RBCs which carries oxygen to tissues. This is because the pair of genes controlling hemoglobin production are defective. Thalassemic person require frequent blood transfusion in order to survive. The thalassemia gene is present on an autosome. Second genetic disorder is hemophilia. Those persons suffering from hemophilia have either a defective gene or lack genes which control production of substance responsible for blood clotting. In the absence of such substance, blood does not coagulate. Once bleeding starts, it does not clot easily. Third genetic disorder is color blindness. Different kinds of color blindness have been detected, but in the most common form of the disorder, a person is unable to distinguish the blue color from green. Again, this is due to the presence of a defective gene or absence of the gene, which are responsible for color vision. The genes for both hemophilia and color blindness are located on X chromosome, and hence the disorder is passed down from mother to the son, because a boy receives the X chromosome from the mother and Y chromosome from the father. In the mother with two X chromosomes, the defect does not show up. Also in the daughter, the effect of defective gene on the X chromosome inherited from mother may be masked by the normal gene on the X chromosome inherited from their father. Since X chromosome bears the defective gene, the son suffers from the genetic disorder, as male has only one X chromosome and one Y chromosome, and so the defective gene does not get masked. So thalassemia is an autosomal genetic disorder, while hemophilia and color blindness are sex chromosomal or X chromosomal disorders. So you will now appreciate why marriages between close relatives termed cousinous marriage are discouraged. So now we move to the next section, that is genetic counseling. In marriages between relatives, chances of inheriting defective genes by the offspring is enhanced as both parents being related may possess the same defective genes. If recessive, the two defective genes expressed when passed on to offspring by both parents. Therefore, it is essential to know the probability of a genetic defect in the offspring and seek the advice of genetic counselors who are professionals. So, genetic counseling helps one to know the chances of inheritance of the genetic disorder so that people can make informed decisions. So, you can well imagine that human beings are complex in structure, behavior and body function. Thus, many genes control the features of that make a human being, that is human genome. It became possible to identify many of these genes of humans, their location on the chromosome and the combination of nucleotides that constitute them. All the various genes together constitute the genome. Knowing the human genome can help in finding and devising therapies for many genetic defects. This promises hope for people suffering from genetic disorder, as the location of every gene of the human genome is now not known. So, genetic engineering, which is also known as recombinant DNA technique, this technique refers gene from an organisms of a species can be transferred to become part of or genome of an organism belonging to another species, which is then termed GMOs, genetically modified organisms. Transfer is possible through plasmid present in bacteria. So, plasmid is a circular DNA molecule found in bacteria. It is not a part of the bacterial chromosome. Transfer is also possible through viruses that attack bacteria in a known bacteriophage, and these carry genes from a cell culture which is grown in a culture dish and transferred into the bacteria. A bacterial clone containing the required gene may then yield the foreign gene, which can be used for replacing a defective gene during gene therapy. So, this is the major steps in genetic engineering. So, we will discuss, we will explain this figure. So, human a restrictant enzyme cuts out a specific segment of DNA from a human cell. This segment usually contains a gene of interest. Insertion into a plasmid. A plasmid is a small cell, circular piece of DNA naturally found in bacteria. The same restriction enzyme is used to cut open the plasmid, creating matching ends. The human DNA segment is inserted into the plasmid forming recombinant DNA. Then transformation into bacteria. The recombinant plasmid is introduced into a bacterial cells. The bacterium now carries both its own DNA and the new human gene. So, then protein production. So, bacterial cell reads the instruction from the human gene. It begins producing a protein coded by that gene, and last outcome is this: the result of a bacterium that acts like a tiny factory making human proteins in large quantities. This is the foundation of biotechnology and genetic engineering used in medicine and industry. This technique allows mass production of life-saving drugs like insulin for diabetes. It is also used in agriculture, pest-related crops, and research studying human genes. So, now we are going to the end, and we will close this program with a few points in the form of what you have learned. So, passing down the characters from parents to children is called hereditary. Children of same parents differ because they possess different combinations of parental genes. These differences are termed variations. We are all human beings but can be distinguished easily from each other due to variation. Heredity and variations are due to genes and their varied combinations. Study of heredity is called genetics. Mendel was the first to postulate laws of inheritance, and he said that heredity was due to factor and that every feature was controlled by a pair of factors which separated into different gametes during gamete formation. Another Mendelian law of inheritance stated that in a pair of genes, one may be dominant and other recessive. The dominant gene of the pair marks the effect of the recessive member of the pair. Sutton found that these Mendelian factors were the genes and that genes were present on chromosomes. So, chromosomes are present in pairs in the nucleus, and each is made up of one molecule of DNA and protein. Diploid number of chromosomes in humans is 46, out of which 22 pairs are autosomes and 2 chromosomes X and Y are sex chromosomes. Genes are made of DNA. They are segment of the DNA molecule of the chromosome. A DNA molecule is a polynucleotide, each of which its nucleotides is made up of a nitrogenous base sugar and phosphate. A DNA molecule is made of two strands of DNA helically coiled around each other. Before cell division, DNA in every chromosome replicates, forming two identical DNA molecules which are present as the two chromatids forming the chromosome. In DNA replication, two strands of a DNA molecule unwind and each acquire a new strand so that two molecules of the DNA are formed. Sex determination in humans is based on combination of sex chromosomes. Females have two X while male possesses one X and one Y chromosome. So defective gene or absence of genes may cause genetic disorder. Example of genetic disorder is thalassemia, hemophilia and color blindness. Thalassemics lack gene responsible for production of hemoglobin, so they need frequent blood transfusion for survival. Hemophilics bleed profusely. Their blood cannot coagulate as they lack genes for factors necessary for blood clotting. Hence, their bleeding does not stop easily. So, this is all about hereditary. We will come again to meet your new program of science and technology. Till then, goodbye and thank you.